This is the Geisel School of Medicine at Dartmouth, and in Lebanon, New Hampshire, in the United States. And just to let you know who have not yet sent it in, we have people registered today from all around the world. I'm just going to read off a couple countries, or a bunch of countries, sorry. So from Canada, Taiwan, South Korea, India, Egypt outside of Cairo, Hungary, Norway, Brazil, India, China. I'm skipping all those from the United States because there's so many people from the United States. Colombia, Turkey, Nepal, Brazil, Spain, India, Slovakia, Pakistan, Belgium, trying not to repeat, Iran, Japan, Taiwan, France, Italy, the Netherlands, Greece, Austria, United Kingdom, Romania, Sudan, Iraq, Germany, I think I said Taiwan already, but Turkey, Brazil, Sudan again, China, India, Mexico, Japan, Italy, Iraq, Mexico, Hungary, Pakistan, Italy, France, Italy, Netherlands, Taiwan, South Korea, Sri Lanka, Tunisia, Argentina, South Korea, Germany, Iran, Brazil, China, South Korea, Kazakhstan, Saudi Arabia. Let's see, any new ones I have missed? Czech Republic, Netherlands, Croatia, France. So some of those are repeated, and tons of people from the United States have registered. So here we have someone else, the Joplin Independent Community-Based Internet News Source headquartered in Missouri, USA. So yes, if you can find the chat box and you're just coming online, we're just sharing where we're located around the world. So find the chat box, find my name, Dawn Van Dam, and send me a quick message and tell me where you're located around the world. And while you're doing that, I'm going to get started because it's the top of the hour. So welcome to 1 Million Solutions in Health and our signature square presentation around a fabulous new technology for cancer vaccines. So I'm just going to give you a little bit of background about us at 1 Million Solutions in Health, and then I will turn the presentation over to our speaker for today. So the agenda is to do a welcome and introductions, and as I said, give you a bit of background about us, have you do the signature square presentation, and very importantly, a signature square presentation is an opportunity for you as a listener to give us really important feedback for this speaker, and for the speaker to learn what you're looking for as they move forward in their science. And without your participation in the signature square and you're filling out the survey, we really can't continue to offer these because it's a really important requirement for us to move forward, is that we get your feedback. So we will shortly be putting up, or maybe we'll put it up now, the survey that we'd like you to fill out. So you can always open that and start to fill it out and give us your feedback as we go along. So at 1 Million Solutions in Health, our goal is to improve healthcare solutions, or improve healthcare one solution at a time. And we do that across the board in life sciences and in healthcare and for patients and consumers. And really our goal is to help shape healthcare by sharing those solutions and accelerate the adoption and the evaluation and the validation of those new scientific discoveries. And how we do that is in a couple major ways. We help people connect across those three areas, life sciences, healthcare, and with patients and consumers. We help learn and share by sharing knowledge, holding events like these, and then also live events. And really what's very important is we'd love you to send us your ideas and dialogue with us about how we can help you share your ideas around the world. Oh, and the last piece is that you can also participate in a collaborative project that we do through our Technology Evaluation Consortium. So the Technology Evaluation Consortium is actually a collaborative partnership within the, actually it started within the pharmaceutical and biotech industry, but now we're expanding it to medical devices, to diagnostics, and to health data solutions. So this is a really exciting consortium that we've had in place for 10 years, actually, and we're just bringing it out to the rest of the marketplace now. So our mission at 1 Million Solutions in Health through the Technology Evaluation Consortium is to triage and streamline new technology consideration for the industry, and be up the place for industry to conduct their evaluations, validations, and be kind of like a funnel or a flow-through way to get the best science moving forward and get science to have the feedback that it needs. That's why, again, I'll just remind you that we're really going to need you to fill out that survey later on. So what we call this comprehensively is we call it our Hidden Gems program. And we call it GEMS because we really want you to get the edge, whether you're a scientist or a technology provider or you're someone in the industry or in academia. We fundamentally have created a marketplace for science. And so we want to help people find those hidden gems through our Hidden Gems program and sort of access what we're calling the secret vaults of science, because there's lots of people out there with great science, but they just don't have a chance to have a voice or have their information shared. Publications are a great way to share information in science and news, but actually getting the science out there through an interactive way, like a webinar like we're doing today, is really important. So fundamentally, we have tons of technology flow opportunities. So we get ideas and recommendations from the industry or specific requests for searches. So if you're an industry person and you want us to help search for new science, we can do that for you. And then we have this special partnership with the NIH where we're bringing you today's science. We also have access into academia around the world, but if you're in academia and like us to help you share your, if you're in the technology transfer office, let's say, and you'd like us to help share your science, give us a call about that. And then we have a database of our own of 2 million plus people around the world who are entrepreneurs and other areas where we can bring science forward. And really, we're trying to get it all right to the patient faster, but getting it to the patient faster means it has to go through the industry and to have them bring it to the marketplace. So that's the fundamental story behind our Hidden Gems program, and we'd love you to be a participant in that. So the Technology Evaluation Consortium is really a subcomponent of that, where we, as it says here on the one side, we bring the end users, whether they be in pharmaceutical industry and biotech or some other particular industry users, and we bring to them the technology vendor or scientists as we are today. And so we sit there in the middle as the Technology Evaluation Consortium and 1 Million Solutions in Health and help bring the science forward. So here's just a smattering of examples of people from the industry, high-level people who have been part of our team over the years or who are currently part of our team, just to get you to look at their titles, probably more than anything else, to tell you that they're very important people who you probably want to share your science with, and they're part of our consortium that we have. So our process is really simple. We look out to find new scientific solutions. We gather input to optimize those scientific solutions, and then we share the findings. So again, it's pretty straightforward, but if you want to know more, give us a shout. So the signature square that we're doing today is going to be very exciting, but I want to just give you a little bit of background on how this works. So really, the signature square is you look at the science, you give us some feedback, and you give us some ratings, and you give us some comments. And as I said before, this is really a critical component of what we do. And so right now in the chat box, you will see a link to a survey through SurveyMonkey, and we'd love you to open that up and fill it out either while you're listening or right after the presentation. It is an evaluation that in the end tells the scientist a whole bunch of information about the market readiness and the value proposition to the market. And we provide back a report to the scientist or the vendor as to what you said, but all your comments will be anonymous. So you can say whatever you like. We will not attribute anything to you specifically. So please feel free to be as direct and honest as you possibly can. And in the end, the signature square produces a result. The science is rated either a blockbuster, an upstart, a novel, or an innovator. So that's kind of why it's called the signature square. The benefits to the industry is it gives everybody a chance to provide feedback. It gives a chance to kind of benchmark each scientific opportunity versus other things that have been put forward. And it gives you an opportunity to learn more about scientific technologies that are out there. And really our end goal, and particularly my passion for 1 Million Solutions in Health, is to really expedite and accelerate the adoption of these new technologies. So by spreading the information around more globally, it's a chance to again, get more people involved. For the NIH, what they're looking for, very importantly, is more licensing agreements. So we're hoping that you're going to listen to this presentation today and say, This is an opportunity for me to license this technology into my company if you are from the industry. They're also looking for more creatives or cooperative research and development agreements. And then the last point is what's coming through in the signature square, the feedback and direction for their future path and input into their roadmap. So it's all pretty straightforward, and we're really glad to have you participating today. So I'm going to jump in and give you a brief summary of today's presentation from our speaker. And so just to summarize what we're going to be covering today, this presentation is going to feature an exceptional discovery in the search for unique cancer antigens. Dr. Arya Bira has isolated a tumor-associated antigen, TAA, that is widely expressed in various human tumors. This sets the stage for a cancer vaccine solution. A new patent will be issued on this technology actually very soon, in fact. Importantly, this TAA contains a number of immune-dominant regions, epitopes, that are readily recognized by human T cells. It has been shown that humans can efficiently elicit specific helper and cytolytic T cells capable of killing human tumors. The focus is to understand how immune cells control diseases like cancer and how we can use this knowledge to develop potent therapeutic formulations to combat cancer. Cancer strategies often don't cure patients, and patients often relapse. To elicit long-lasting disease-free survival, current thinking suggests the need to induce immune responses against cancer. In considering a cancer vaccine, in order to be effective, it needs to have a unique target against which immune cells will be responding. So with no further ado, I'm going to introduce our speaker and then turn things over to him. So one moment, and I'll just tell you a little bit about his background. So Dr. Arya Biragan, sorry, I'm used to calling him Arya, received his PhD from the Institute of Molecular Biology at Engelgart Academy of Sciences of Russia, Moscow in 1991. He obtained postdoctoral training from the University of Illinois at Urbana and the National Cancer Institute. He was a scientist at the Science International Applications Corporation in Frederick, Maryland for about 4 or 5 years, where he worked on the development of new generation therapeutic vaccines for B-cell lymphoma. In 2000, he moved to the Vaccine Biology Section, National Cancer Institute as a scientist, where he continued his cancer vaccine studies. He was a tenure-track investigator at the Laboratory of Immunology, and he is a tenured senior investigator at the Laboratory of Molecular Biology and Immunology, National Institute on Aging, and also an adjunct associate professor at the Department of Molecular Microbiology and Immunology at Johns Hopkins University. He has received various awards in the past, including a Staff Recognition Award at National Institute on Aging and a tenure promotion at National Institutes of Health. He currently has 4 patents, excluding 6 others that are pending. So with no further ado, I'm going to just tell you a couple things. One is, please, if you have any questions during the presentation, please put them in the chat box to me, and I will ask them to Arya. And also a reminder to please fill out the survey either during the presentation or after the presentation, and as we need your feedback. So here we go. And we just need to mute. There we go.
Okay. Do you hear me?
Yep, perfectly now.
Okay.
Over to you.
Okay. Good morning or good evening, depending where you are. So first of all, I'm honored to have this presentation, and thank you very much to Dawn Van Dam for organizing this presentation. My name is Arya Biragan. Also, I'm known as Bira Arya. I'm senior investigator and chief of a section called Immunoregulation at the National Institute on Aging. Okay. So one interest I'm pursuing for many, many years is to understand tumor biology in order to develop strategies to combat with this deadly disease. One way to do is to use vaccines. You all know about vaccines to infectious diseases, to parasites, and even to influenza vaccine. Probably some of you already have been vaccinated by a seasonal influenza vaccine. However, these vaccines quite significantly differ from cancer vaccines, because vaccines to infectious diseases is to induce immune responses against something foreign, which is what our body is designed to fight with xenogenic and foreign things, and they do it very well. For example, if you engraft foreign tissues or cells into our body, immune cells very efficiently eliminate them. In contrast, cancer, you may think as an uncontrolled growth of something or part of our own body or tissues or cells. And therefore, immune system, which is very potent in eliminating various impaired cells and so on, they are not well engaged in killing cancer cells, because cancer cells use a very potent machinery which protects us from autoimmune diseases, such as employing directly or indirectly immune cells by themselves to control other immune cells. And therefore, the challenge for cancer vaccine is how to induce specific immune responses against cancer cells or cancer itself without harming us, without inducing autoimmune diseases. In order to do that, we need a unique marker. In other words, it's also called tumor-associated antigen, which is only expressed in tumor cells, but not in other cells. And therefore, if you use tumor-associated antigens, one can envisage to activate specific immune responses, recognizing these tumor-associated antigens. It means cancer cells which express or produce these antigens and eliminate these cancer cells. However, as a self or as something, part of our own body, tumor-associated antigens often poorly immunogenic. And therefore, there are many, many ways people use to render them immunogenic. And we, for example, developed a very simple strategy, for example, to imitate infection by using chemokine-linked tumor-associated antigens, about which I'll briefly talk a little later. So, once you have a unique antigen or tumor-associated antigen, which is only expressed in tumors, there are many, many applications, and I'm only listing few of them. For example, you can develop cancer vaccine, and as I said, cancer vaccine is designed to only eliminate and kill cancer cells without harming other tissues and cells. Or you can, one branch of or one spin-off of cancer vaccines is dendritic cell vaccine, which is you can pass dendritic cells, which is very important antigen-presenting cells, with your tumor-associated antigen or pieces of this antigen, and then infuse back into patient. And you know that there are many, many clinical trials using this approach in humans. And another application is cancer diagnostics. Just knowing that unique tumor-associated antigen is expressed in certain cancers, you can do certain diagnostics, or even to use as a predictive marker for disease-free survival after various vaccines and various interventions. So, and as I briefly mentioned, you can use unique tumor-associated antigen expressed in cancer to generate potent vaccine formulation against cancer. And as I said, that often tumor-associated antigens are very poorly immunogenic. It means if you take just tumor-associated antigen and inject back into host from which you generated this antigen, the chances that host won't induce any immune response is very high. And therefore, one way we circumvented this problem by linking tumor-associated antigens with chemokines. And chemokine is a small peptide, is designed to recruit specific type of immune cells. And the idea was to not only bring towards the vaccine site specific type of immune cells, but also to let this antigen to be internalized or uptaken by immune cells of our interest, so they would eventually induce immune responses. And I'm just showing you just one representative picture on curing B-cell lymphoma in mice. It's immunocompetent mice where if you challenge them with B-cell lymphoma, mice usually succumb to this lymphoma and die within 20 days. However, if you treat with our vaccine, then you can get at least 60% disease-free survival. So, I showed you that the proof of principle that if you have a specific tumor-associated antigen, you can at least cure tumors in mice. However, there is a very big need to find a unique tumor-associated antigen which could be used in humans. And you probably know about many, many antigens. I'll talk to you about one antigen. It's called SpanXP. Actually, real name is very, very long, so I am just, for simplicity, would say it's SpanXP. This is a very small protein, about 108 amino acids long, and only in healthy tissues it is expressed in spermatozoids, in sperm. What we did is that we cloned this antigen, and also we synthesized various peptides to analyze whether they are immunogenic in humans. So, SpanXP, first, I'll tell you what we found. SpanXP is widely expressed in many, many human tumors, which is very important thing. And also importantly, I'll show you data that SpanXP is immunogenic in humans. So you can induce immune responses, and very important immune responses are so-called cellular immune responses, which is induction of T helper CD4 responses and cytolytic CD8 T cell responses, which eventually should eliminate cancer cells expressing SpanXP. So this slide to show you, just this is a representative slide to show you that SpanXP is not expressed in normal tissues and cells. We tested many, many tissues from healthy people, and you can see SpanXP is not expressed in none of them. And it's many variety tissues. However, SpanXP abundantly expressed in human tumor cell lines. Just this is example to show you that melanoma cell line expresses abundantly this antigen, SpanXP, as you can see with this brown color. But this is just cell line. How about primary tumors? If you test in biopsies from human tumors, you can see melanoma, lung cancer, ovarian cancer, breast cancer, colon cancer, and also I'm not showing you here non-small cell lung cancer, and so on and so on. All of them express abundantly this antigen, tumor-associated antigen. And very interestingly, when we tested more specifically in melanoma, we found that actually its expression more associated with metastatic state of a cancer. You know that tumors divided into various stages, stage 1, stage 2, and 3, and so on. And then stage 3 and 4 is most deadly form of a cancer, when cancer is metastasized in various organs. And when we did an analysis of expression of SpanXP, we found that it is mostly expressed at the metastatic stage of a melanoma. And this finding recently was corroborated by others, showing that actually SpanXP is mostly expressed at so-called epithelial mesenchymal transition stage of cells, which usually, in other words, associates with the metastatic stage of tumor cells. So, in order for any antigen to be effective as a cancer vaccine, it needs to be able to induce T helper responses, more specifically T helper Th1 type of responses, and also cytolytic CD8 T cell responses. So to test it, we used peripheral blood T cells from healthy humans, and as you can see, SpanXP protein, either it's protein or various immunodominant regions of SpanXP, which we call peptide 5 and peptide G1 and so on, all of them very nicely induce Th type 1 helper CD4 T cell responses, as shown by expression of interferon gamma in these CD4 cells. More importantly, SpanXP protein or its immunodominant regions induce cytolytic CD8 T cell responses in human T cells, as shown here by this representative slide. As you can see, these CD8 cells from humans raised against SpanXP can nicely kill melanoma cell lines, for example, as you can see here, or model cell lines transfected with SpanXP. But more importantly, if we take a biopsy melanoma cells from patients, this slide shows that SpanXP CD8 T cells, which we induced ex vivo, nicely kill melanoma cells, patient melanoma cells, in HLA-A2 dependent manner. One issue with SpanXP is that it is only expressed in higher primates and in humans, but it is not expressed in any other animals. And therefore, trouble is that SpanXP cannot be evaluated as if, in mice, because if you inject SpanXP into mice, it'll be foreign to mice, and of course, it will induce immune responses, such as antibody and T cell responses in mice. And this just shows a slide that we actually using this mouse model. Nevertheless, we're showing that we defined B cell dominant regions in SpanXP. So However, to demonstrate you that even though SpanXP is a human self-tumor antigen, you can actually use it to induce very potent immune responses by using another tumor-associated antigen. It's called oncofetal antigen. Unlike SpanXP, this oncofetal antigen is expressed both in humans and mice. I mean, in malignant cells of humans and mice naturally. So, and therefore, this self-tumor antigen for mice can be used to validate vaccine responses and to extrapolate whether you would induce similar things using SpanXP. And as I previously told you that if you inject oncofetal antigen into mice, it won't induce any responses in mice. However, using our strategy, which is linking with chemokines, such as in this case, so-called MIP3 alpha, or another name is CCL20, if you link this antigen with oncofetal antigen and inject into immunocompetent biopsy mice using DNA, so-called DNA vaccine, you can induce cytolytic CD8 responses, as you can see from this slide, which only kills tumor cells, mouse tumor cells, that naturally express OFA, as you can see, A20 lymphoma, but not other other tumor cells. And importantly, if you use our vaccine strategies with OFA, actually you can eradicate or eliminate tumor. It's actually, we used the treatment in mice which already had a bulky tumor, and we treated them with this vaccine formulation. As you can see, it nicely eliminates, induces tumor-free survival. So these results indicate that not only self-tumor antigens can be used nicely in humans to induce immune responses, which eradicates tumor-associated antigen-expressing tumors. Also, I showed you the data of a very unique antigen. It's called SpanXP. And important thing is that this antigen is expressed in numerous human malignancies. And therefore, one can envisage to create a single vaccine formulation to treat various human malignancies. And I showed you that data that SpanXP also mostly expressed at the metastatic stage of melanoma. And this is very important, as you know that tumor by itself doesn't kill humans, and it can be removed. But main problem is how to combat this metastatic metastasis. And therefore, SpanXP based vaccine probably could also eliminate metastatic cells. And also I showed you that SpanXP is a very immunogenic in humans. It can induce potent T helper CD4 responses and importantly, cytolytic CD8 T cells, which can kill tumor cells expressing SpanXP. So, and I showed you that just using SpanXP or defined by us peptides of SpanXP or immunodominant fragments of SpanXP, you can actually generate, using human healthy T cells, immune cells, which can recognize tumor cells and kill. And all these findings are included in our patent application, which will be issued this year in March. And finally, also I briefly talked about our vaccine strategy, which is very simple, versatile, and potent, just linking antigens, tumor-associated antigens, with our with any tumor-associated antigen, linking with chemokines, you can elicit potent therapeutic anti-tumor immunity. And you can either use SpanXP or even OFA as a cancer vaccine in this application. And this technology we also applied for patenting. So what applications can be used for SpanXP? So for example, as I said, you can generate cancer vaccines using either our technology, such as chemoattractant or chemokine-based cancer vaccines. Another thing is that you can directly pulse dendritic cells of a patient with SpanXP peptides, which we defined, to elicit potent anti-tumor immune responses. And as you know, dendritic cell vaccine trials are being tested in many, many cancer patients currently. Or you can use SpanXP as a diagnostics tool. Since it is mostly expressed at a metastatic stage and also in many variety of cancers, you can do using antibodies, which we have, or your own antibody raised against SpanXP, or so-called RT-PCR method to diagnose patients or potential patients for cancer or for residual cancer. Also, SpanXP can be used as a predictor for disease-free survival after various interventions to combat this cancer. Lastly, if you need anything about science-wise, you may directly contact me. Here is my number and email, or you can find me on web. Or for legal technology transfer matters, you can contact Joseph Conrad or Betty Tang from Office of Technology Transfer of NIH. And thank you very much for your attention.
Thank you so much, Arya. That was fabulous, your presentation. We're just going to flip the slides back over to me, and I'm going to start to ask you some questions that have come in from our attendees today. So thank you, everyone, for attending. And just a reminder, in the chat box is the survey for the signature square. And I just want to remind everyone to please fill it out immediately, because feedback is really, really critical for this process. So I'm going to start here with a couple questions. So has this technique, and sometimes I'll ask you a question that maybe has been answered already, but you might want to actually expand on it. So I just ask them all. So has this technique been used in pancreatic adenocarcinoma?
So you mean whether this our finding is applicable for treatment of pancreatic cancers?
Sure. Yes.
Yes, we see. We actually our findings we reported, and SpanXP is also expressed in pancreatic cancers, and therefore, as any cancer, you can use it to induce immune responses against pancreatic cancer. However, one should really this cancer vaccine is designed to be effective at the minimal residual disease. So let's say after various modalities, interventions with pancreatic cancer, once a patient is recovered and the immune system a little bit restored, then you have to use cancer vaccines for example, based on our SpanXP. And cancer vaccine is designed to eliminate residual disease, which escaped from various current modalities.
Great, thank you. We had a request here, and maybe I'll just explain. The way the NIH works is because we're looking to have more licensing and collaboration agreements, and just we want to track the success of this program, we would like everyone to go through Joe Conrad. And so if you could contact him, and then he will set up a time for you to meet or talk to Arya. So we have here someone that said, excellent and exciting work. Can you please show your contact information again, please? Thank you. So if I'll leave that up for a few moments so people can copy it down. But if you could contact Joe, that would be great. And then we'll get the ball rolling, so to speak. So Dr. Beer, here's a question for you again. In the mouse model, OFA beyond 60 days still resulted in about 20 to 40% mortality. How can one read into the data? Does the observation have any implication to clinical trials?
Okay, so let me answer this way. Actually, this we published data. For example, if you observe these mice for longer, once mice are protected, it's a long-lasting immunity. And therefore, one way to test it, you can re-challenge with the same tumor. Then same tumor doesn't grow. However, one should be very cautious. The tumor is heterogeneous. Even this, the lymphoma, what we are using, or any human tumor, they are heterogeneous. And therefore, whenever you eliminate tumor, either a tumor contained, for example, some mutated OFA, or, for example, or some cells which do not express OFA, that can actually give a rise to tumor. On the other hand, tumor also mutates to eliminate, this pressure from immune system or from vaccine. So all these things are main challenge. However, so far at present moment, of course, cancer vaccine is the only so far modality or treatment modality which can eliminate drug resistance or which can eliminate residual tumor. And then, there is also even thinking that during that elimination, immune cell can attack other tumor-associated antigens. It can besides your vaccine. And therefore, the answer to your question is that once a tumor is eliminated in this mouse model, which is very difficult model, this is BALP-C with A20. If whoever worked, it's almost very few people or labs could treat them and get this type of results. And therefore, you can extrapolate that even though mice are not little humans, you may get a similar efficacy in humans.
Great. Okay, I'm going to give you one comment, and then I'm going to move on to a whole bunch more questions that have come in. And just a reminder, if you have any questions, just find my name, Dawn Van Dam, and stick it in the chat box. Thanks. So one person here says, I'm really enjoying this wonderful presentation. Thank you very much for giving us this opportunity to learn and share. So thank you for that. That's great. So a question for you. Does SpanXB presence have genetic or epigenetic origin?
So about epigenetic origin, we didn't test. And certainly it is only, and no one knows what is function of SpanXB. It's actually itself scientifically very interesting. But surprisingly, it is expressed only in spermatozoids, and people speculate why it is in spermatozoids. But in cancers, it is very well expressed, and why it is expressed, we do not know at present moment.
Okay. so this one might be very related. Can the expression of SPANX be detected by mapping the person's genome so that preventive measures can be taken to prevent malignancy?
No, my answer probably would be no, because you need to test expression such as mRNA or protein. By genomic analysis, of course, it would be sitting there, but not expressed.
Okay. Here's another question. Does SPANX-B have any relationship with another antigen called SPANX-C?
Yes, this is a very good question. You know, there is another antigen also expressed in higher primates, and which is also in sperm, very closely related. It's called SPANX-C. However, SPANX-C, even though it's related, it doesn't have a, at least it differs in some stretches of amino acids there. And as you know, probably, SPANX-C also considered a good tumor-associated antigen. Some regions of SPANX-B overlap with SPANX-C, and therefore one can envisage just using SPANX-B, you can cover also SPANX-C. In that sense, they are homologous, but SPANX-B contains additional important regions which elicit immune responses.
Okay, great, thank you. Can SPANX-B be utilized together with other antigens?
Oh yes, this current, many even clinical trials, people use several tumor-associated antigens to increase, coverage. In that sense, it is yes, it can be used. And here we're only showing that SPANX-B alone can do certain important responses, but it can be, and therefore used with combination with many other modalities and antigens.
Okay, great, thank you. Here's another question. Can you please explain your chemokine-based vaccine strategy?
Okay, this is actually, we have 3 patents. I didn't show these references, but you can contact to our OTT office for those. And these are, before when we created, people, even now, people use antigen together with virus adjuvants and so on and so on. But idea what we wanted is to imitate inflammation. If you want to inject into skin, can we induce some kind of imitate inflammation? And then one way, what we wanted is that we actually wanted to recruit important immune cells, targeted way, not only to recruit, but also to specifically deliver antigen into those recruited immune cells. So your vaccine won't be diluted by being uptaken by any unneeded cells. And we tested in many, and we published in many of papers in Nature Biotechnology, in Blood, and so on, all this mechanism, how it works. It turned out that it's such a simple strategy. You don't need any whatsoever adjuvant. You just need to link with chemokine. And why chemokine? Chemokines, there are 18 different chemokines. They target, let's say, 10 different receptors. And these chemokine receptors uniquely expressed on immune cells. And therefore, one can design a formulation which only targets certain type of immune cells. And that we tested, and it showed that it is truly very, very potent strategy. And important thing, surprisingly, even though antigen what we were using is so-called self-antigen, itself it doesn't do anything. Just linking them with chemokines, it induced very good immune responses. And then important thing here to emphasize that chemokine should be physically linked with antigen.
Okay, thank you. So
Sure.
a bunch more questions have come in. So again, I'll remind people if you have a question, just stick it in the chat box, please. So does expression of Spanic B change with aging?
Oh, that's a very good question. We have no clue. We honestly, this is a beautiful question, but we never tested. But just screening these normal human tissues, which spans, various ages, people, so far we don't see its expression in any normal tissues. And except, of course, in a head of spermatozoids.
Okay, thank you.
Sure.
Another question here. Does Spanic B vaccine induce autoimmune disorder side effects, such as sarcoidosis seen with ipilimumab prescription?
So this is an excellent question. But since we don't have any model where Spanic B naturally expressed unless you test in humans or in primates, higher primates, there is no possibility to definitively answer. However, just logic and our tests indicate that since Spanic B is not expressed in normal tissues, there is no possibility of induction of autoimmune diseases to any other cells, normal cells, because they don't express Spanic B. And Spanic B, in autoimmunity, one way of autoimmunity is induction specific T cell responses which recognize only tissues or cells expressing the target antigen. Since this target antigen is not in those tissues expressed, there is therefore probably there won't be any autoimmunity.
Okay, thank you.
Sure.
This one I'll ask you and let you see if there's an answer for it. So what are the potential adverse events?
Oh, I can only speculate. For example, potential adverse effect would be loss of sperm cells, and therefore, but since most of cancers usually occur when we are beyond our reproductive age, I think loss of sperm may not be a big issue. Besides that, I don't even envisage, as I explained about autoimmunity to previous question, I don't envisage any harmful autoimmune responses, unlike any other tumor-associated antigens being tested currently in humans. And also, we don't see any harmful effects even when we tested in mice with OFA vaccine. So far, answer would be probably not. No harmful effects.
Okay, great. Now, I know you're trying to look for collaborators and partners to bring this to market. So maybe you have some thoughts about this next question. Can you tell me more about the type of formulation the vaccine is available in or would be available in, whether it is nano-emulsion or liposome-based?
So, we did not do many other strategies. We did not test other vaccine strategies. And one can envisage. Here is one most important thing is that the target antigen, if you have a target good antigen, which covers many malignancies, human malignancies, and which is not expressed in human normal cells and tissues, that's the most important thing. And then if this antigen can elicit T-cell responses, specifically cytolytic CD8 responses, one can envisage to use any other modality to induce immune responses, whether it's liposome-based or whatever, or even dendritic cell vaccines, whatever currently you have. Or we can help you to develop this chemokine-based also vaccine strategy that's very, very simple. It's unbelievably simple, or any other, as a matter of fact, vaccine modality.
Great, thank you.
Sure.
So I know, again, that you're trying to look for partners to help you bring this to the market, but has Hispanic B at all yet been tested in any humans?
No, I'm our lab and our interest is, to work on the basics. Basics, what it means. We prove of principle. We prove that certain things is very interesting and so on. And then primary our goal is to publish high-impact papers, which we did. And this Hispanic B was published in Clinical Cancer Research. It's a very good journal and so on. But by publishing that, not only I get a good grades, good rating, but more importantly that we hope that other people would see it and then use it. So, and therefore, the goal was to mainly to create strategy and to create know-how, and then other people can take it further. And therefore, no one yet used it for clinical trial, and it's up to grabs, I would say.
Yeah. So just a few other questions have come in that are kind of related to that. So just to clarify, right now there are no ongoing clinical trials with Hispanic B, and so therefore it has not yet been tested in humans. So even your points about whether or not it has any adverse events, you're just speculating because we don't know. And even your point about whether or not it might have any influence on the reproductive cycle or whether it should be used in, could be used in males or females who are younger, we don't know. So those are all right now unanswered questions. You were just kind of hypothesizing, correct?
That's right. But my hypothesis is not just a pure hypothesis. It is based on a sort of knowledge, on a knowledge how autoimmune responses occur and so on. And since, and another point for this unique antigen is that since it is for cancer, which mostly develops when we stopped our reproduction, let's say, then even if it is used and it affects sperm production, I believe it won't cause big harm. But that's, of course, it's a sort of hypothesis.
Right. And I guess we're talking about cancer, and cancer kills. And if
yeah.
you have a cancer vaccine that can be universally utilized across all types of cancers, at least a person could make a choice. Do I want my cancer to be gone and therefore I live, or do I want my cancer to perhaps be at risk of not being gone just for the chance that maybe I want to have another child or something like that?
Exactly, and one should also consider this. This is cancer vaccines. Majority of now trend is that it's supposed to be used at the minimal residual disease. When one or patient got various treatments and so on, and then usually cancer relapses. And at that stage, after treatment, when cancer is at the minimal residual disease, and when cancer, various cancer cells hidden stay, only way is to use vaccine to eliminate them. It means to use our own body's immune system to find those hidden cancer cells and eliminate. And for that purpose, cancer vaccine is a very good strategy.
Yeah, the way I liken it, just to make it really simple, is, when I cut myself, my body heals myself, right?
Yes.
My white blood cells race there, I get a scab, it eventually heals over, and I'm back to normal again. So basically what you're saying is this would allow my own body to kill my own cancer cells, which would be pretty amazing, right?
Yes.
So I'm just going to ask Joe Conrad, who's actually on the line, to unmute himself and just say a few words. And I'll just, while I'm doing that, I'll just see if anyone else has any more questions. Please put them in the chat box, and I'll wrap up with those after Joe has unmuted himself and he chats. And so there you go. There's Joe.
Hi, everybody. Thank you, Dawn, and thank you, Aria, for doing such an excellent presentation. I think the attendees were astonished is not the right word, but I'm very happy to hear about this very excellent technology that's coming out of the NIH and that's available for further development. So to the earlier question, has this been studied in humans yet? That opportunity is wide open through collaborations, through create opportunities, collaborative opportunities as well, and even licensing from potential companies that want to move this to the patient population that it's targeted for. So I want to thank everybody for attending. If you do have further interest in this technology and you'd like further information or you'd like to actually engage in some type of collaborative relationship with the NIH for this particular technology, don't hesitate to contact me. And what I will do is I will put you in contact with Betty Tong, who is the licensing specialist for this technology. I believe she's also on the webinar as well. But as the coordinator of the activities or the interactions with Dawn's organization and the NIH, we'd like to have all of those inquiries funneled through me first before I pass them on to Betty, so we can actually track them and make sure that we have your contact information and everything that we need to further the engagement. And so, again, NIH is looking for partners to help move this to a patient population that desperately needs it. The reason we do this is primarily because the NIH is in the business of discovery. We are not in the business of manufacturing. So we need our corporate partners, our industry partners, and university partners as well to help move this technology out of the NIH into the private sector so that the patients can actually get the benefit of what it is we have created. So I'm looking forward to hearing from as many of you as possible. And with that, thank you again. I'd like to turn it back over to Dawn.
Yeah, I'll just ask one more question to you, Aria, that has just come in. So I'm not sure this is your expertise, but how long would you estimate it would be before this might see clinical application in the most optimal state?
So I honestly, it's a very difficult question I can speculate many things. So one thing is that from a characterization point of view of a SpanXP, it's almost done. Not almost, it's finished. So it's ready to go and to be tested. But what it takes to go to clinic is that, again, it depends on a person's facility, whether they are already ready to go to start applying for IRB and so on, all those things, and whether the vaccine formulation, how they are going to use with SpanXP is ready to go. And therefore, it probably probably around 3 to 5 years to do phase 1 clinical trial. If everything goes well and if FDA approves the trial and so on, it can go, depending on one's facility and ability to go. Yeah.
Great. So the challenge to everyone on the line today is call everyone you know who is interested in bringing cancer vaccines to market and get them to call Joe Conrad, and let's get this one to help millions and billions of patients all around the world and eliminate this disease of cancer. So I'm just going to remind everyone again one last time to fill out the survey. If you click on it now while it's in the chat box, it'll still be available for you to fill it out even after the presentation's over. So I would, again, extremely encourage you to do that. And again, I want to thank everyone for their participation today. And again, I'll leave it to all of you to spread the word and help us get this to the market. So thanks again to Dr. Aria, and thank you to everyone who has attended.
Oh, thank you very much.
Our pleasure. And we hope to hear more from you in the near future, which I think we will. Take care, everyone.
Thank you.
Have a great day. Bye now.
Okay. Bye-bye.